Yeah. Okay. Today we deal with another class of biological motors, which are myosins. And myo means muscle, right? And whenever we think about the human body structure, the first thing that comes to our mind is the or when we talk about the beauty of the human body, we think about the muscles, right? Because they generate the force for our movement, for things that are essential for our life. When we move about, when we are eating, or even when we are talking, that's the muscles that are at work. So it was always a great fascination to humankind, that what exactly is this muscle. Now, so here you can see a microscopic image of a muscle, and what you see are these alternate light and dark bands, which are called striations. And this is what was observed by Anton van Leeuwen. Yes? So this is what was observed by Anton von Leeuwenhoek when he was looking at objects under his microscope. So Anton von Leeuwenhoek is kind of credited to have begun the whole field of cell biology, because he was the first, not the first, but maybe one of the first persons to be actually looking at objects under a microscope and trying to understand what they look like. So at this time in the late 1670s, 80s, that's the time a lot of new ideas came forth because of some technological advances. And what was that technological advance? It was the advancement in the way glass was being ground to produce lenses. So that helped people understand how the stars, or what the configuration of the solar system is. So people were able to make observations, telescopic observations, and they were also able to look at microscopic objects, for example, cells. So here, Leeuwenhoek actually looked at this dried muscle fiber, and he was able to understand that there is some sort of periodicity to it. Right? There is a periodicity. That's what he was able to find, and to him, what was apparent was that or there is something in this muscle which is not two-dimensional, but has a cylindrical three-dimensional shape. So that much he was able to understand. So so this is 1680s, right? We are talking next about 1860s. So almost 200 years later, people were again trying to understand what this muscle was. And then there was an observation made by Leon Frederick, where he said that, okay, before that, these striations were named as bands. So for example, this dark band, you could call it the A band, and the light band, you could call it the I band. And what Frederick observed was that when the muscle contracts, the A band does not vary much, but what varies is the I band, that is the light bands. They seem to contract, and then they seem to go back to the original position. So the contraction and of the muscle coincided with the contraction of these I bands. But at that time, there was practically no idea what these were. We now know that when we are talking about these muscle fibers and we are talking about these striations, what we are actually looking at are bundles of actin filaments, which are cross-linked by something called myosin motors. So this is again a structure of this same muscle that you were looking at here. This is at a lower resolution. If you look at it at a higher resolution, this is what you get to see. And these dark bands actually correspond to something like this. So here, And if you kind of look at the cartoonic depiction, then this is what it turns into. So this kind of muscle that we are talking about is the muscles that you would see maybe in your arms or your legs, called striated muscles. And what happens is that during formation of these muscles, the muscle cells fuse together to give you these long filamentous cells. And each of these cells have these enormous structures of actin and myosin. So here is what you would call these muscle cells, which are showing you these structures of actin and myosin. And you zoom into one of them, like you zoom into one particular region, and you will see something like this. And this is the cartoonic depiction of that. So what do you see here? You see parallel arrays of actin, right? You see parallel arrays of actin. So here, each of these lines are actually parallel arrays of actin. So each of these are parallel arrays of actin. And they seem to originate from one point, which they named as Z line, so which is here. So you have parallel arrays of actin, and then you have them originating from one point. All right. Now, in between them, in between the parallel rays of actin, you have these myosin filaments, cross-linked myosin filaments, which appear as these dark ranges here. So these are your cross-linked myosin filaments. Now, what about the A band and the I band then? Something that Frederick had observed, that the A bands remain almost invariant, but the I band shows contraction every time there is muscle contraction. So you think of this as repeat units now. And what happens during muscle contraction is that because of the action of these motors, these actin filaments slide inwards. So when they slide inwards, if you have these actin filaments sliding inwards, then you would have a reduction in the space that is occupied by this I band, or just this part of the muscle, just the actin part. So this is the I band that contracts every time there is muscle contraction, and then goes back to its position when the muscle relaxes. So this much is kind of known to us, or something, what I've said so far is not something new to you. It's already known. You already know that this is how muscle contraction works, right? You've learned this in your school. But this was not so evident for people who were trying to understand how muscle works. Initially, people thought, because a lot of things turned out to be helices, right? You had DNA double helix, you had collagen, which was a helix. So a lot of things were helices. And you can stretch a helix, right? If there is a helical conformation, you can stretch it, you can relax it, much like a spring. So at one point, people even thought that maybe these muscle myosins or the actins or whatever there is actually acting like a spring, and the contraction and relaxation of the muscle is nothing but the contraction and relaxation of the spring. And it turned out that with advancement of technology, with advancement in biophysical techniques such as X-ray diffraction imaging or cryoimaging, or even force measurements, for example, optical trapping measurements, it became clear that this is not the case, that it is not like a spring, but there is some other mechanism behind it. Okay, so what we have seen so far is that this muscle contains something called the thick filament. Again, the thick filament, the thicker ones here correspond to these myosin. Then you have these thin filaments, which are actin. And then you have these regions where you would see just the thick filaments and the thin filaments together, which would form the A band, and then a region where you see only the thin filaments, which are the I band, and the I band is one that would that shortened, and that is what Frederick had observed, right? And then now we know that it's not kind of a spring-like action, it's not contraction and relaxation of the spring, but rather what happens is that you have sliding of these filaments. And the whole idea is based on the sliding filament hypothesis, and that which has now been proved right. Okay, so we go back to St. Yogi, and we had already seen how Yogi and his team were instrumental in identifying actin as a component in muscles. They had isolated actin, and they had come up with one of the most fundamental observations to this actin myosin theory, to this actin myosin field, and that is if you take actin and if you take actin, then it is going to form a very viscous solution. But if you take actin myosin now together and you add ATP to it, you will find that the viscosity of the solution decreases. So you take actin myosin, it forms kind of a gel with a certain viscosity. You add ATP and the viscosity reduces. And we can now understand that this reduction in viscosity should have something to do with contraction, because that would enable this change in viscosity. So there is something to do with ATP that plays into the way myosins work, not just how ATP polymerizes, but also into how myosins work. Now, here we come into a cartoonic depiction of what a myosin unit looks like. Like we had seen what is the subunit of actin. We had seen that it is one monomeric subunit that keeps polymerizing, and then you have this entire filament of actin being formed. We had seen how tubulin polymerizes. We've seen that the alpha-beta tubulin dimer is actually the polymerizing unit and not an alpha and a beta and then an alpha. It doesn't work like that. You have an alpha-beta dimer, and then successive addition of these dimers gives rise to formation of the microtubule filaments. Here in myosin, the primary unit that polymerizes doesn't have one polypeptide, doesn't have two polypeptide, but seems to have 6 polypeptide chains. Okay, this is a general structure of this myosin. And what you see is that everything seems to be in duplicate. So you have these two blue structures, you have these two red structures, and these two yellow structures. So if you just look at this image, you would see that there is a helicity in this. So it's again something that resembles a filament, right? This part would resemble something that is a filament, and you can think easily how this might give rise to a filament like this, because if you have filaments, then you might be able to take this one filament and wrap it around another filament, and you can give rise to polymers in this way. So that can be envisioned. But what about this structure? What about the structure that is projecting out of this filament? So this structure, which is here called the head structure, is actually the globular region of these myosins. And the globular region has this ATP binding site, and it has the actin binding site. So both these properties that are crucial for the functioning of myosin, that is binding to ATP hydrolysis, and binding to actin, are both attributed to this head. Whereas this structure, this helical structure, is attributed towards formation of I wouldn't say polymers, but exactly oligomeric units. So multiple myosin units would come together and form this kind of filaments, thick filaments, with the help of this helical tail unit. So that's about the simple geometry of the myosins. But what about the red and the blue? So it turns out that there are some additional proteins that associate with the myosin, and together they actually form the polymerizing unit of myosin. And these are called the essential light chain and the regulatory light chain. Why light chain? Because if you take myosin and do you know what an SDS page is? Before I go further, Ratnesh, do you know what an
Yes, ma'am.
SDS page is?
Yes, ma'am.
Okay,
SDS page
Piyush,
is an
Piyush, you know what an SDS page is?
Yes, ma'am.
Okay, so if you take myosin and if you run it on an SDS page, you will not see one protein, but you will see three proteins. So you will see one protein which is of very high molecular weight. So you call that a heavy chain. And you see two proteins which are of considerably smaller molecular weight, something about 1920 kilodalton. The heavy chain would be 200 kilodaltons plus. So those small chains would be called light chain. The reason for calling them heavy and light is just this. You run it on an SDS page, you see that they have different molecular weights, different sizes, vastly different sizes. All right. So historical perspective. So you have something called a light chain, you have the heavy chain, and there are two light chains, and one is called the essential light chain, one is the regulatory light chain. What their functions are, and the rest of it will come in a bit. So how do we know that this is the structure of myosin? I mean, how exactly do we know? So initially there were clues. So initial clues came from electron microscopic observations, of course. You put the myosin under a TEM or transmission electron microscope and you will see structures like this. So you see a helical tail and you will see these globular heads, and you understand that by now you've understood that these globular heads are required for both ATP hydrolysis as well as binding to actin. What you do not see here very clearly are these light chain regions, but if you could somehow sort of stain this myosin by some means only for these light chains, you would see them occurring here. So let me give you an analogy. So they call this region where these light chains and regulatory light chain and essential light chain binds as the neck region. Why neck? Because you have this filamentous tail, which is kind of stiff. I will not say it's very stiff, but it's kind of stiff, right? And it can polymerize with other helices. So it's like you take one helical wire and you take another helical wire and bring together, and it would form another helix. And you have these two globular heads jetting out of those helices. But if you have to give the globular head some degree of flexibility to be moving around, then you need the linker between the head and this tail to have flexibility. So what you could do is you could say attach. So you take a helical wire and you take a ball, and between the helical wire and the ball you attach kind of a spring so that the head or the ball kind of moves around, blobs around. That's exactly what is done by the helical region that is present, the neck region that is present in the myosin. So if we go back here, this region, this region which they just named it here as neck, the neck region where the light chains bind, provides a degree of flexibility for the heads to move around, for these myosin head regions to move around. And you can actually understand the flexibility if you look very carefully at these images. So you look at this image and you compare it with this image, you can see that there is a degree of flexibility by which this head domain has actually moved a little. So this dynamism is provided by the presence of this neck region. You delete that neck region and your myosins will be very stiff. Okay, now when people were initially trying to understand what myosins were, they were doing a lot of different experiments with whatever was available to them. So what was available to them at that time was an idea of proteases. So they knew how to digest protein with proteases, just like they knew how to digest DNA and RNA with DNAs and RNAs. So they took these proteases. For example, they took trypsin, and they found that if they add trypsin to myosin, they end up getting two distinct units. So if what it turned out is that if you add trypsin, it digests it somewhere from here, so that it creates two units of myosin, which is called heavy meromyosin and light meromyosin. Now, just to recapitulate, you have already looked at heavy meromyosin when we were looking at the image of an actin filament that is linked to heavy meromyosin and that was viewed under an electron microscope. And that gave the idea of the presence of a barbed end and a pointed end, simply because of the way these heavy meromyosins are positioned along the actin filament. It gave the feel of an arrow. So if this is an arrow, you have a pointed end and a barbed end. And as we have learned that the barbed end is the region when new actin subunits are added. All right, so let's go back now. So they added trypsin and they were able to get two distinct units of myosin. And they found that it was not the light meromyosin, not this region, but this region that was capable of binding actin and ATP hydrolysis. So whatever I've explained so far, that the head domain is the domain that actually binds actin and has the capacity to hydrolyze ATP. This was identified in this way, the very stepwise way. First, they found that, okay, you add actin and myosin together and it forms a gel-like solution. You add ATP and it seems that the gel contracts. So definitely the contraction is dependent on ATP. Then they were able to isolate the myosins and they found that the myosin itself can hydrolyze ATP, that it is an ATP dependent process. Then you want to identify which part of myosin. So what about cleaving myosin? You cleave the myosin now and you have two parts and you try to understand which part is now able to bind actin and cleave or hydrolyze ATP. And you find that, okay, it's the heavy meromyosin. Then of course at that time again there was very little other option but to use other proteases. So they used another protease called papain and they could further cleave this heavy meromyosin into two other subunits. So this part, which is the S1 subunit, was found to be the point where the ATP hydrolysis and actin binding activity both exists. So we come back to the same point that we have highlighted before, that in the polymerizing unit of a myosin, which is actually made up of 6 polypeptide chains, 2 polypeptide chains of heavy subunit and 2 each of essential light chain and regulatory light chain, combined together to give you one polymerizing unit of myosin. And in this polymerizing unit, the heavy chain has a globular head. And that globular head, as identified by this papain cleavage, is the point for both binding to actin as well as ATP hydrolysis. Okay, so here is another classic experiment that showed what was or that helped people understand what was exactly happening with myosin. So what did they do? They took the heavy meromyosin. Which one is the heavy meromyosin? It's this. You just take this part. They took this heavy meromyosin and a lot of experiments at that time were done with heavy meromyosin because they understood that that is the part that binds actin, cleaves ATP, hydrolyzes ATP, and you don't need this region for that matter. So it kind of simplified their experiments. They took this heavy meromyosin and what were they observing? They observed that if you just take the heavy meromyosin and you add ATP, you would find like a very fast hydrolysis of ATP occurring. And this is occurring in the absence of actin. So you don't need actin for this activity. You take heavy meromyosin, you add ATP, and ATP is hydrolyzed almost instantaneously. So this here on the x-axis shows you how many milliseconds. So you can appreciate that within like 2 or 3 milliseconds of addition of ATP, you have hydrolysis of ATP. And then the ATP hydrolysis sort of saturates, right? So you have an instantaneous increase in ATP hydrolysis and then it kind of stops. There is no more ATP that is hydrolyzed. But in this very same system of heavy meromyosin, if you now add actin, then what do you see? You see that, okay, ATP hydrolysis again happens instantaneously. There you add it, you add ATP and ATP is hydrolyzed instantaneously, but it does not saturate and it keeps rising. You see this graph rising upwards. So why was that? So to understand this, let us now look at the way we understand this entire myosin activity working. And so let us now consider that. Let us now consider a few things. For example, the first point is the binding of actin. This activity is present on the head domain, right? That's the first point. The second point is ATP is also hydrolyzed in this head domain. So with considering these 2 points, let us look at this. If you have heavy meromyosin, that is only the head subunit, and you add ATP, the ATP is hydrolyzed, which means let us consider a situation where you have myosin and you have ATP, and the ATP goes and binds to this myosin head in the absence of actin, because that is what we saw, the ATP is being hydrolyzed in the absence of actin, right? So you think of the head unit. So let us consider this as the head unit, right? And it binds to ATP and it can hydrolyze ATP, right? But then once it hydrolyzes ATP and it forms ADP plus PI, the next part is this can now have a weak affinity for actin. So the idea is that as long as it is bound to ATP, it cannot bind to actin. It somehow cannot bind to actin. But when ATP is hydrolyzed to ADP and inorganic phosphate, that is when it has a weak binding affinity to actin. So it can then go and sit on actin. It binds to actin. The next idea is that now you have ADP and inorganic phosphate, and we have seen in multiple cases that binding of ATP is one state. By hydrolysis of ATP giving you ADP and inorganic phosphate is stage 2, and release of phosphate and binding to just ADP is stage 3, right? We've seen that in actin polymerization, we've seen that in microtubule polymerization. Let's put that consideration back here. So you have the head, which is binding to ATP, right? It's hydrolyzed ATP, and now you have ADP plus PI, which has a weak affinity for actin and goes and binds to actin. Now, if the phosphate is released, that is when there's something called a power stroke happens, and there is a change in the conformation. So here the change in conformation is actually all due to this helical neck piece that gives it this flexibility. It's all because of that little bit of flexibility offered by the neck region. And that makes a power stroke, that cocks this entire thing, makes a power stroke. And then it pushes back the actin filament. It's almost like, have you seen rowing? I mean, I don't think anybody of you has done, but if you have done rowing, even better. Have you seen how people row? Now kaise chalate hain? So the first position is where they sit straight with the oar. Then they go forward with the oar and then they push it back. You think of it like that, exactly like that. You have this head that binds to actin, then it cocks it, then pushes back, and then it is released. Now, For this entire thing to happen, what kind of conform or what is driving these conformational changes? The conformational changes are all driven by these different states of ATP. So you have, when it is bound to ATP, it does not bind to actin. When ATP is hydrolyzed to ADP and PI, that is when it has a weak affinity for actin. The release of PI makes a state where it is hooked and it produces that power stroke. Let's go back now to understand this a little better with the help of these diagrams. So you have a state where it is bound to ATP, and this is not the preferred conformation to bind to actin. But as we have seen before, the head itself can bind to ATP and hydrolyze ATP. So that really doesn't change if you add actin. It still binds to ATP and hydrolyzes ATP, which indicates that the binding of to ATP and hydrolysis is not dependent on actin. So it binds to ATP, hydrolyzes ATP, and gives this ADP plus PI, which has a weak affinity to bind to actin. And this is the slow step in this reaction. Now, after this, you have the release of inorganic phosphate, and that creates this power stroke, that creates the change in the conformation, or let's think about as the three-dimensional positioning of this head with respect to this filament. So that changes, creates the power stroke. And the moment ADP is released, you would see that the entire this power stroke is actually executed and finished, and now it has moved into another position. It is slightly better to visualize this with a video, because now it's easier to see things that way. So let us look at this video. Can you hear the explanation that is given with the video? Can you hear that?
No, ma'am, the voice
No.
is not.
Okay, if the voice doesn't work, then I will explain it as it goes. So what you see here is an actin filament, right? This is an actin filament, and we know that actin polymerizes by addition of these monomeric actin units, and it has like, if you look at it from the top, it seems to have a double helical nature, but actually it's repeats of these actin monomers which polymerizes on the barbed end. And here you're looking at a muscle, a myosin polymer, and the head of one. So we know for each polymerizing unit you will have two heads. So we are looking at one of the head just for the sake of visualization. And this here is the linker region, which gives it a flexibility to move around. So this is the head, myosin head, actin filament. And what I would like you to do is to look at the position where the myosin head will bind. So we go, oh, sorry. Okay, so we will go ahead. So you have this actin filament and keep a look at this position. So when it is bound by ATP, as I had explained, it doesn't have affinity for actin, but the myosin head by itself can hydrolyze ATP, right? And the hydrolysis ATP to ADP plus PI gives it a weak affinity. So you have ADP and phosphate, and you have, okay, so you have these two heads, and one of them is going to be binding to actin. You see this? And ADP plus, yeah, that is the state where it is bound. Now, release of this phosphate causes a conformational change. So uh, you have so what did we see? We saw that there is a conformational change at the time when the inorganic phosphate is released. Let's have a look at this movie once more. So when it is bound to ATP, it does not have an affinity for actin. It is hydrolyzed. by the ATPase activity of the head domain, give you ATP plus PI. This has a weak effective actin, and this head now seeks out actin at the point where it can contact actin. There is a particular conformation where it binds or on the filament. And this binding also induces a certain conformational change, which helps in release of this containing phosphate. So once the containing phosphate is released, that is when it causes a change in the way this globular head is positioned with respect to this filament. So you see, this is what is called the power stroke. So you have this motion which is creating a and now the head. So now the next step is that the ADP is exchanged for an ATP, and every time ATP binds, you know that it doesn't have an affinity for actin, and so actin is released. So you have conformational change, and you see every time it's actually pushing the filament a little forward. Every time this works, it pushes the filament a little forward. Look at the push. So in effect, this is actually a very beautiful system by which myosins work. Now, so far, do you have any questions? Yes? I cannot hear you. Maybe you can write down your question. You can write it on the chat on the side. No? Okay. Or maybe you can try again. Anybody else has any questions? Is it one step movement for every PI released? Yes. Yes. That is what gives something called a step size to this entire process. And I'll come to this, or we will come to this in the next class, where we are trying to understand how exactly can you analyze what is called a step size. So the whole idea is that it doesn't work like a spring. It's not that the muscle contracts and relaxes by a spring action. Then it would have been a continuous action, right? Rather, these are discrete steps which are working. So every time there is a PI released, you have one movement of actin. But if you think of a muscle filament, you do not have one subunit head acting on it, not even two from one polymerizing unit, but multiple polymerizing unit, therefore multiple heads attached to it. Therefore, it is a coordinated effort of all of these individual heads and not one head. So you would see the pull or the push, whatever it is, being that much more. So you would see the force rising very high. Because if you think of one motor head, the amount of force it can generate is in a few piconewtons, just like polymerization of microtubules or polymerization of actin. But again, if you think of actin or myosin polymerization, actin or microtubule polymerization, then we know that if we combine a lot of these filaments, then we can increase the force generated. It's the same thing here. If you combine a lot of these motor head actions, you can increase the total force generated. All right. So, okay. Let's move on and try to understand a little bit about this actin binding site, etc. this barbed end and the pointed end, right? And you might have a question that, okay, when the myosin binds to the actin filament, then towards which end, or how does the binding of this myosin work with respect to the actin filament in terms of the barbed and the pointed end? Because we know that actin, so when we are talking about this, we are actually not talking about the exact muscle thing that we have talked about so far. So let me bring it into context a little bit. So we know that actin myosin is present in muscles. But if you look at an individual cell in our body, then that cell is also moving. And you would be surprised to know that you also have muscles, myosins in those cells. That does not mean that those cells are muscle cells, right? So you have non-muscle myosins too. You have myosins, you have different kinds of myosins. You have myosins that make up the actin myosin complex present in our muscle cells. You have other myosins that are present inside other cells that help those cells to move around. So for example, if the cells needs to divide and constrict, so at the point where the cell divides, there is an actin ring formed, right? But as the cell constricts and forms these two daughter cells, during this point, there must be some forces that helps this ring to contract, right? You have a big ring और उसको छोटा बनाते जाना है आपको. तो ये छोटा बनाना कैसे हो सकता है, कैसे हो सकता है? If you have a spontaneous depolymerization happening of the actin at this point, then maybe this is not the ideal case. So what's happening at this point is you have myosins actin on this actin, and that brings this ring to constrict because you have sliding of one filament against the another filament, and you can constrict this ring. Similarly, when a cell is moving and you need to coordinate the movement, one way is you can have polymerization of actin at the ends, but another way is to also help in sliding of these actin filaments. So these sliding motion can actually bring the cells to move together. So kind of an amoeboid movement, think of it. And you would understand that an amoeba doesn't have a very smooth movement, right? It's not that it can only be actin polymerization, तो बहुत smooth होगा. But amoeboid movement is kind of looking around for its base, right? That's what macrophages do. That's what cells in our body that have amoeboid movement and are looking for those small bacteria that it can eat up. So उसके लिए क्या होना चाहिए? You should have some movement of these actin filaments, and that is what is done by these myosins. Think about that, then the binding of this head onto the actin filament and its movement should also have some sort of directionality, right? Do you think it should have a directionality to it? And if you think it has a directionality, do you have any clue which direction it could go? Positive end, negative end?
And positive end?
Yes, it does go towards the positive end. What happens is that, again, just to simplify this whole thing for you, think of a person who's rowing a boat. Think of a person who's rowing a boat, and you have the end of this sort of how do I explain this? So you have a person who's rowing a boat, and the movement is like this, right? So every time it moves, what happens is that it is pushing the water backward, but itself the boat is moving forward. So with the rows, what it does is, with the oars, what it does is it's trying to move the water back, and thereby it propels it. So the myosin head also does something like this. It sits on the actin, and it kind of, and every time it cocks, when there is the release of PI, there is the formation of the power stroke, and then the ADP is released, and you have this movement, and then ATP binds to it, and then it detaches, right? And so every time this happens, it moves it sort of moves the filament in one direction, and it itself seems to be keep moving in the other. So what happens is, relative to the filament, there is a new position at which the myosin binds. So every time it moves the filament, it might be in the same position. It need not move, but what it is doing is, with every step it is sort of moving the actin filament, and it's creating a new site for it to bind on the actin filament. Okay, so again, what Sarika had asked is very crucial here. There is a step size to it. That means that there is a particular orientation in the filament where it can bind, and that orientation is periodic. Why? Because you have a periodicity to the way the actin monomers are arranged on the filament, right? And it has been found that the step size is about 10 nanometers. We'll come to greater details about this in the next class. For today, I'll just give a briefing that it was found that this step size is about 10 nanometers. There were multiple people trying to analyze it. Some said 60, some said something else. Finally, it was found to be 10 nanometers, and that's the period. And why is this periodic? It's periodic because you have periodicity in the way the actin monomers are arranged. So every time it will search for another site to bind, and that site has a periodicity in this filament of about 10 nanometers. Therefore, ये bind करके जब move करता है, दूसरा जब आता है, that's about 10 nanometers. So that's where it would bind. So this initial binding to the actin filament, the point it seeks out that binding region, is the slow step in the reaction. That is where it takes time for this process to occur, and that point is also the point where the myosin has a weak affinity for actin. That's also very important. It has a weak affinity for actin. All right, so we come to this important paper that was published in 1993. So before that, people did not quite have an idea. So how do you now, so these are the very fundamental things. So how do you attribute that a certain say function is present at some distinct part of a protein? You understand that by, as I showed you, you can digest a protein and then look at which part of that protein has that function, right? How do you understand that a certain residue or a certain amino acid is required for the functioning of the protein? You can mutate that residue, and then you can see if that function is retained or the function is lost, right? How do you understand how a protein actually binds, or what is the conformation of a protein when it binds to a certain protein or it is in a certain or in a certain active state? How do you understand that? You need to look at its crystal structure, or you need to have some sort of an understanding of the structure of that protein. So that is what happened in 1993 with publication of this crystal structure. And what it shows is this S1 fragment. So going back, what was our S1 fragment? Our S1 fragment was this head that had a little bit of this neck region bound to the essential and the regulatory light chains. So here is your actin. Okay, so let me point out which one is the essential light chain and which one is the regulatory light chain. The rest of it is all this myosin head region and the neck region. So yellow is the essential light chain here, and this in purple is the regulatory light chain. So you see there is a distinct way by which the essential and the regulatory light chains bind, and they bind specifically to this neck region. And the rest of it here is the globular head that we were discussing so far, and here is the neck region, which is the flexible region that helps it to undergo these power stroke conformations. And in this you have an actin binding site here, so the site that is actually seeking out actin, and right opposite to that you have an ATP. Right opposite to that here you have a site where ATP hydrolysis is taking place. So ATP hydrolysis at this site would make the actin binding site somewhat open to binding to actin. So this heavy, the heavy myosin chain, the head domain, has these two sides. So you have one side which is seeking out actin, and this conformation, the conformation of this site sort of changes when there is ATP binding and hydrolysis at the opposite end ATP binding site. And the whole thing is translated into changing the position of this head in terms of this liver. So this lever arm is holding the head in place, and it's only a change in conformation. And the change in conformation is almost to the degree of 45 degrees. So when there is the release of inorganic phosphate, the position of the head with respect to the lever arm in the pre-power stroke and the power stroke state is almost 45 degrees. So it's a very sharp change in angle between these two regions. All right, so in muscles, you would not see actin being bound with myosin all the time, because there is something called a troponin-tropomyosin complex that is present on the actin. So here is a beautiful cryo-electron image of actin in complex with tropomyosin. Now, what is tropomyosin? Tropomyosin is again a polymeric filament. So it's two filaments which are intertwined together, and the structure looks more like this. So here is your actin filament, and this yellow thing is actually tropomyosin. And tropomyosin also has a helical conformation, and its helical periodicity is longer than the actin helical periodicity. And it wraps around actin in this manner. So what happens is that the region where actin is, where myosin binds to the actin filament is actually hidden, because tropomyosin binds to those regions, and it doesn't allow myosin to bind to actin in those regions. Now this, I'm saying, is very specific to muscles, because in the rest of the, in muscle cells, you are not looking at polymerization, depolymerization events of actin. It's there. If you find fast polymerization and depolymerization of actin, then the muscle, the whole structure of muscle would collapse. Rather, it's very steady and it's there. So there must be a way to regulate it. The way it is regulated there is by having this layer of tropomyosin around it, which blocks binding of myosin to actin, because it's bound to the same place where myosin would bind. So then how does it work? So it turns out, so you can see here, you can clearly understand how it would look. So here is your actin, and this sort of caging around it is the tropomyosin. It turns out that the tropomyosin also has something called so you have something called I get so confused with this. Troponin tropomyosin. So troponin, when it is bound to increased calciums in muscle cells, much like in all the rest of the cells in our body, the calcium is stored in endoplasmic reticulum. And there is a special endoplasmic reticulum in muscles which stores very high levels of calcium. So when muscle cells are excited, you have release of calcium from the sarcomeres. Now, this release of calcium actually causes a change in conformation of this tropomyosin, because the troponin calcium complex changes the way the tropomyosin binds. But this change is very little. What happens is, at that point, the change is enough for myosins to push out tropomyosin from the actin filament just a little bit, so that then myosin can bind to it and cause this filament sliding motion. So what we've seen so far is that in muscle cells, there is a way to regulate how and when myosins can bind to actin. There is a certain periodicity to it, and it is linked to the cycle of ATP hydrolysis and release of PI and release of ADP and binding of another ATP. And we have seen that the actin filaments in muscle cells are bound to this tropomyosin, and tropomyosin binds to the same, or it kind of covers the grooves where myosin binds, so that you need to sort of move this tropomyosin just a little bit for the myosins to come and bind to actin. And this little motion is caused by an increased levels of calcium in the cytoplasm. And this occurs because if you can excite the muscle cells, so when the nerves gives it the stimulus and the muscles are excited, that's when the calcium is released, and increased calcium levels then causes the calcium troponin tropomyosin complex to actually move just a little bit so that myosin can bind, and it further pushes out the tropomyosin from the actin. But the whole thing is still bound to actin. And so when the calcium levels go down, it can go back to the conformation where myosins can no longer bind to actin. So with this, I will stop the class today. So it was just an introduction to myosins. In the next class, we will look into details of how the step sizes were measured so that you have an idea of how the forces are built up by myosins inside a cell. So far, do you have any questions? Okay. Just stop sharing and I'll take any questions if you have. This was just an introduction to myosin, very brief introduction, but it introduces two very important concepts. One is of the dependence on ATP, and that ATP hydrolysis is not dependent on binding to actin. That ATP hydrolysis is an inherent property of the muscle myosin head, and the head domains have an ATPase activity, so they spontaneously hydrolyze ATP, and that this is possible even in the absence of actin. But what happens is that once the ATP is hydrolyzed and gives rise to ADP plus PI, that's when the myosin heads have a weak affinity to actin. And so this whole thing is also occurring in non-muscle cells, where non-muscle myosins also show this phenotype. But in muscle cells, the actin is specifically covered by this troponin tropomyosin complex, so that the myosins cannot always bind to actin. You need elevated calcium levels for that troponin tropomyosin complex to move a little bit so that it reveals the actin binding sites, myosin binding sites on the actin, and the myosins can then bind to actin, where the head is actually bound by ADP plus PI, weak affinity. And then this binding causes a conformational change, the inorganic phosphate is released, and that causes a cocking. So that causes a 45-degree angle change in the head with respect to the rest of the filament, the neck. And when it cocks, it's actually kind of moving this entire filament forward. It causes that power stroke. Then the ADP is released, ATP binds to it, binding of ATP completes the cycle, and the myosin head detaches. ATP is hydrolyzed again, ADP plus PI, another binding, another power stroke, PI released, out again. So this is just a very simplistic explanation of how myosin works, but it took almost, I don't know, from the point of finding out myosins and actins in 1940s to the point where they came up with the first image of the step sizes was in 1990s. So almost 50 years. All right, I'll stay here for 5 minutes if you have any questions.
Ma'am,
Yes, come on.
actually, ma'am, the function of the regulatory chain and the function of the light chain is to give the flexibility to the whole structure. So why they are named differently, like too heavy, too essential, and too regulatory?
Okay, essential. So, essential chain is sort of seen as something that kind of keeps this conformation of the head and the neck together. But the regulatory light chain has some other function. And I didn't say it today, but I can just give it in brief. So this is how you think of myosin, right? You have the two heads, you have the helical tail, and think of my wrist as the neck. है ना?
Yes.
So the idea is that this is not how it stays in solution. And it has been found that it actually sort of, folds onto itself. I can't do it, but with my hands. But if you try to imagine this, it kind of folds and sort of doesn't have the kind of conformation like this, but kind of folds down. So for this folded down conformation, where the heads are positioned in a way कि वो ऐसे है and it's not looking up, that's not the correct conformation for it to be seeking out actin. So for the change in the conformation, there are some sites on the regulatory light chains that are phosphorylated. And they're phosphorylated by upstream kinases. So here comes the entire event of signal transduction, where you get a signal from the extracellular environment, and it can then regulate a number of kinases. Those kinases and even those downstream signalings can do multiple things. One, it can induce active conformations of WASP, activating ARP, activating actin polymerization. Second, it can induce the kinase activity of downstream kinases that can act on myosins by activating the myosin regulatory light chains by phosphorylating them and helping the myosin get out of this conformation where the heads are down, folded down, and go up like this. So this conformational change is regulated by the phosphorylation of some residues on regulatory light chain. So that's the function of the regulatory light chain. The essential light chain sort of keeps this entire structure in place, because if you remove the let me get back to this in the next class.
Yes, ma'am. Sure.
But this is more or less what it is: the essential light chain and the regulatory light. The regulatory light chain has sites which regulate the way these heads are positioned with respect to the entire with respect to the entire filaments. So generally they are not like this. This is how we like to think of them. They are like this. But if they are always like this, they would always be trying to seek out actin filaments. So you can sort of push them down and have them below so that they are not seeking out actin filaments. Then how do you push them back up again? That is done by regulatory light chains.
Okay. Thank you.
So we'll come to this again in the next class, where we will look at how signal transduction events are doing this. So multiple signal transduction events have if you have studied signal transduction, you would have read how one signal can actually elicit a number of different processes. So it has a bifurcation. But they can all integrate into one single event. So if you have a signal transduction that is giving rise to changes in actin polymerization properties, for example, giving rise to barbed ends, right? That same event, somewhere in that event, you can also have increased in kinase activities of enzymes or kinases that can actually act on those regulatory light chains and make myosin more active. So not only are you branching actin, but you're also increasing the sliding movement of the actin. Net effect is that the cell is moving forward. And it is a directed movement because you're creating branches at one specific end, because you've increased the activity of wasp at one end of the cell, not throughout the cell. So everything is regulated in this way. Simplistic view again.
Yes, ma'am.
Ma'am, I have a question as well.
Yes, Farman.
So, like, must be having something that is so, but as we have studied,
I think
actins are
your
very
voice is breaking because
dynamically
I
unstable.
could not hear
So,
your
how these
question well.
actin remain static? Ma'am, am I audible now?
You're audible. Yes, go ahead.
So, ma'am, sarcomere must remain very static in their structure, right? Like they cannot, their conformation cannot be changed. But actins are very dynamically unstable. So, how these actin maintain their structure in sarcomere so that the entire structure remain stiff, like
Ah,
that only?
a very good question. Very good question. कितने एक्टिन एक्टिन आइसोफॉर्म्स है मैं पहले आपसे पूछती हूँ आपने पढ़ा है कि एक्टिन आइसोफॉर्म्स
Uh, yes, ma'am, maybe alpha, beta, gamma. Uh,
तो इनमें से कौन सा वाला मसल में है मैंने ये एक बार
mm,
मेंशन किया है एक्टिन जब बढ़ा रही थी तब
yes,
मसल
ma'am.
में इनमें से कौन सा एक्टिन है हाँ
Alpha, ma'am, alpha,
ओके
alpha.
अल्फा राइट अल्फा एक्टिन है अभी ये आइसोफॉर्म्स जो है ये एग्जैक्टली isoforms इसलिए है because they resemble in their structure, right? They have kind of the same conformation, everything, but they have some inherent differences in the way they act, in the way they have their polymerization depolymerization rates in presence of divalent cations. तो हर चीज हमें यहाँ पे देखना है कि इनका पॉलिमराइजेशन डिपॉलिमराइजेशन कैसे होता है इन प्रेजेंस ऑफ डाइवेलेंट कैटायंस व्हाई बिकॉज एज आई जस्ट नाउ एक्सप्लेन डाइवेलेंट कैटायंस यहाँ पे बहुत सारे काम करते हैं एक तो एक्टिन पॉलिमराइजेशन में भी हेल्प करते हैं दूसरा है कि कैसे उनके साथ मायोसिन बाइंड करता है उसको भी रेगुलेट करता है तो मल्टीपल पॉइंट्स को रेगुलेट करता है एंड इट्स फाउंड दैट alpha actin in general you would not be able to depolymerize it the way you depolymerize beta or gamma actin.
Okay,
So your answer lies in the way the alpha actin, it's a specific isomer of actin which you find in these muscle cells. And that has a different way by which it depolymerizes and a different rate of depolymerization in presence of these divalent cations compared to beta and gamma.
okay.
So
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 alpha,
the so
alpha
the whole
isoform. Yes, ma'am. So alpha isoform persists a longer range of time for polymerization or depolymerization, right?
because the depolymerization kinetics is very, very slow in presence of these divalent cations for alpha.
Okay,
For the alpha variant. So the alpha isomer is the one that's the reason you have a different isomer for actin in specifically in muscle cells. You do not have one very specific to kidney, you do not have one very specific to liver cells, you have or fat cells, you have it very specific for muscle cells because of the property of this isoform being different from the others because of the property of depolymerization in presence of divalent cations. I'm mentioning this repeatedly. In presence of divalent cations, this is what they had found that the properties of depolymerization is different for alpha actin compared to beta and gamma.
okay, ma'am. And ma'am, also, which book should we refer to this, like
See,
studying this
take
subject?
any standard cell biology textbook. I have mentioned one. I'm always referring to one. You take anything. You take Cooper, Bruce Alberts, anything. You take any one standard textbook and you read it. It will give you an idea about actin, myosin, all of these that I'm talking about. I
Okay,
might be referring to some papers here and there, and those papers I'm referring to you to give you a historical perspective. जैसे मैंने आपको कह दिया कि ये एक्टिन बाइंड करता है लेकिन एक्टिन बाउंड स्टेट में सॉरी ये एटीपी को बाइंड करता है लेकिन एटीपी बाउंड स्टेट में ये एक्टिन को बाइंड नहीं करता क्या प्रूफ है ना आप पूछेंगे क्या प्रूफ है तो इसीलिए फॉर हिस्टोरिकल पर्सपेक्टिव्स आई एम ब्रिंगिंग आउट सम पेपर्स to show you that this is how they came to this conclusion from time to time, not for all but for some. I'm giving you a historical perspective, but whatever I'm saying is present in any standard cell biology textbook. I'm not saying anything more than that.
okay, ma'am.
All right, students, thank you so much. So we'll meet again next Friday because tomorrow is a day off, and I think Saturday is a working day, right?
Yes,
Please
ma'am.
cross check. I think Saturday is a working day and we have a class on Saturday. Even if we do not have a class on Saturday, I will request a class on Saturday because we need to have a few classes.
Yes, ma'am. This Saturday is working Saturday.
Saturday is working Saturday. Perfect. Then we meet on Saturday and next class is on Friday.
Yes.
on MS Teams. Thank you so much. Thank you. Bye bye.